Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Today on the Microscopists, Paul Verkade from University of Bristol talks about what inspired him right back at the start to develop correlated imaging methodologies.
That certain structures, like tubular endosomes, they fall apart upon chemical fixation. So that's where I came up with the idea that we need something that we can do live imaging, because those tubules are not there all the time, and we need to be able to freeze that.
He talks about co-establishing the Wolfson Bioimaging Facility.
Together with Mark Jepson, we set up the Wolfson Bioimaging Facility, probably one of the very first really integrated light and electron microscopy facilities.
And spills the beans on his favourite Christmas movie.
What's his name? Hugh Grant and...
Love Actually.
Love Actually. I still cry with that.
Don't think I still cry with it, but I still watch it every year. All in this episode of the Microscopists. Hi, I'm Peter O'Toole from University of York, and today on the Microscopists, I'm joined by Paul Verkade from University of Bristol. Paul, how are you today?
Not too bad, thank you. Yeah, it's been I've done an experiment today. That's been a long time.
In the lab or just
in the lab? Yeah, I've been in the lab today.
Okay, so now I'm utterly intrigued to know exactly what you were doing in the lab.
What do you think? We were trying to do correlative microscopy.
Yeah, but you said you were doing the experiment. Surely you weren't just on the microscope.
Every now and then I do go to the microscope. This was the preparation. Tomorrow we'll go actually to the microscope. So
proper science, proper
science. Yeah, it doesn't happen too often anymore, unfortunately.
You sent me one picture. Well, you sent me a few pictures, but the one picture. Now you said you've been in the lab. This is you in the early days.
Definitely.
I'm looking at the picture of you, and you look a lot younger.
I was a lot younger. That must be at least 15 years ago. I'd probably just moved to Bristol, so that was 2006.
And with your Leica rapid freeze, what was it called? MPAC 2 plus RTS, the rapid transfer system. And I guess the rapid transfer system was the special thing about it. Yeah. My claim to fame is where you could wheel this machine right next to any light microscope, confocal, do live imaging, take your sample, put it into that high pressure freeze, and it's frozen, and then we can process that. So really doing that correlative workflow on live imaging. So critically, you're actually looking at what's going on inside a live cell, and within a second, it's frozen.
Yes. The machine takes about 2.5 seconds. We have about 1 second. So let's 3.5 seconds is what we said would be the time resolution. We were still pretty good because we couldn't do that before.
So critically, if things are moving in the cell and you want to correlate it in an electron microscope, that enables you to use that live cell light microscope, freeze it really, really fast, such that things haven't really moved much. So you can capture exactly what the ultrastructure of where that is going. Well, how did you... Conceptually, it sounds dead easy. For a microscope, thinking of taking something from one microscope and getting onto another microscope. Actually, this is not so simple. So how did you think of it? How?
Well, it started off around 2000 because there were two seminal papers by Roman Polishuk, Alberto Luini, and Sasha Mironov. Those were using, at that point, they were using GFP, but they were using special imaging dishes. And the imaging dish always has a glass bottom, but this glass bottom had a finer pattern engraved in it. So that allows you to see the pattern in the light microscope, but because it stands out a little bit,
it's also imprinted into your EPOM block. And that's basically where it started off. I always had the idea that that was done with chemical fixation, that certain structures, like tubular endosomes, they fall apart upon chemical fixation. So that's where I came up with the idea that we need something that we can do live imaging, because those tubules are not there all the time, and we need to be able to freeze that. And that's where, at a certain point, I had a discussion with the head of the facilities. At that time, that was in Dresden, at the Max Planck Institute for Molecular Cell Biology, with someone called Ivan Baines. And we decided we'll call Leica and ask them if they're interested in that idea. And that's where we called up someone called Ian Lemswood, very good friend now. And he had almost hung up until he, all of a sudden, I don't know, he saw the light of, oh, maybe this is a nice idea. And that's where we started developing that machine together. And that was really, yeah, one of the most exciting things, that I've done, I think.
I think it's amazing the company was so willing to work in partnership and to develop it through, because it doesn't often happen. Not at that early concept stage, not at the concept stage, even not just early, at the concept.
Yeah, no, exactly. That's where it would have been cool to have basically get the picture that I gave them was a picture of a light microscope, a black box, and a high pressure freezer. That was the whole concept at that point. And that's where we developed it from.
That's difficult, but you're not an engineer in background.
Yep.
So Paul, what was your first degree in?
Sorry? I actually am a qualified teacher, so that's why I had one of the other pictures in there. So I'm Dutch, as you probably know. There, the education system is a bit different. So if you want to become a teacher, that is a separate degree. That's not a university degree. Mm-hmm. And so when I was 17, I hadn't got a clue what I wanted to do. But I did like biology, and I did like chemistry at school. And so that's where I decided, okay, I'll go and do a teacher's degree in chemistry and biology. And I really enjoyed that, not from the start, because at the very first, it was probably one of the very first days in that setting where we had to give a presentation. And my presentation at that point didn't make much of an impact, because my feedback was, oh, did you present as well? We didn't notice. So I was done so fast that nobody had the time to make any notes. So that's where I then went on, and then you go into schools to practice.
Yeah.
And like I said, I was 17, 18 at that time. I was cycling into school, and then some of my students came in by car, who were also 18. They were just as old as I was, and I had to teach them. That's why I realized maybe at the moment I'm too young to do that, and maybe this is not my so to say career. And that's why I decided, okay, you could then transition from the teacher's education into university, and that's why I decided, okay, I'll start doing biology. So that's in Utrecht. That's why I studied.
I've got to say, this picture, I presume, is not you as a teacher.
That is not me as a teacher. So there's a few things I can talk about. This is Chris Neal. And Chris Neal,
like a lecturer, as you'd expect a lecturer, with a wooden cane and a mortar hat and black gown and.
Yeah. So this is, like I said, this is Chris Neal. He's one of the technical experts in our Wolfson Bioimaging Facility. But he also plays in a Pink Floyd tribute band. And he dresses up, he's a background singer, but he always dresses up as characters from certain songs. And I assume you know now which song this is.
Oh, yes. Yeah, it's already going through my head.
Yes. So that's where I really love that. Yeah.
So it's a nice cyclic loop, isn't it? The teaching is all the way there, and we'll come to courses later on. But you said you didn't know what you wanted to do when you were 17. So I've never asked you this. Of all the times when you were a child, what is the first job that you can remember thinking, I'd like to be a...
I have a very bad memory.
Ooh.
I really can't remember. No.
So you can't remember any of the jobs you wanted to be when you were a child?
No.
Never wanted to be a football player?
Yeah, but that's. I was thinking something more realistic. That's where I'm going to deviate a bit from your question, because I think so. My dad was a carpenter. He's long retired, and I was the first one to go to university of the whole family, and my grandparents were really against it, because they thought that's not what our people do. They don't go to university, and that's why I'm really grateful and thankful for my parents. They defended that and said, no, he decides his own future, and that's what I've done ever since. It's where, yeah, sometimes opportunities just appear, and that's probably what happened throughout, yeah, my life is chance, opportunities.
So your dad was a carpenter, did you say?
Yeah.
So was mine.
Yeah. So that's where
he could do almost everything in the building trade.
Yeah, definitely.
How's your carpentry skills?
Okay. We've just changed last weekend. The weekend before, we changed all the door handles in our house. So.
And no swearing or cursing when you were doing it?
No, that can't be repeated here.
Yeah. See, my carpentry skills, I think I'd disappoint my dad hugely.
Yeah.
He's exceptionally good, and I'm exceptionally poor at anything DIY.
Yeah. No, I try to be, but I will never match those skills. That was, yeah.
I comfort myself that at least I can use a microscope, and I don't think my dad can. So,
yeah, exactly. Yeah,
we have things. So, from your degree, can you remember what the first microscope was that you used, light or electron?
Electron. Yeah, because already during my undergraduates, I did two projects. One was molecular genetics, and one was electron microscopy. So it was at the time when molecular genetics was really hot, and where basically I was escorted by one of the postdocs to get 1 microliter of restriction enzyme, which was locked away, and I didn't like it that much. And then I did the electron microscopy project, and that really was where, yeah, this is what I find exciting. This is what floats my boat, and that's what it's been ever since. Yeah.
Remember what the microscope was?
That was at that time a Philips. I don't know the model. Yeah.
Oh, can't remember the model. Wow.
Yeah, I know that the second one I used, it was a 420. So, and after that came the CM100, CM200. But that's where the 420 was special. So that's when I was doing my PhD with someone called Arie Verkade, who was quite a famous name in the Netherlands. And that Philips 420 was really a special microscope. Only certain people were allowed on there. And after 2 years, I was allowed on there as well. So I was really proud of myself yeah. I made that progression.
So from that, so that's where you found your passion. You're now you went to Bristol and you have an academic career. You're a professor now at Bristol, but you also set up and were heavily involved in the core facility side as well at the time when you set it up.
Yep.
Which side did you prefer, the academic or the facilities side?
The experimental side of the facility.
Okay.
Running a facility itself is not what is really exciting, but working with other people, developing new protocols, doing fun stuff, or at least for me, fun stuff is what excites me. So it could be on plants, tissue, cell cultures, but it's the technical question or the technical solution for a biological question that I like working on. That is really what excited me.
So, Paul, excuse my ignorance, Paul, I should know better. But, I see you as you collaborate with so many people.
Yeah.
So you're sort of probably more academic now than facility in your job description, your core. But is your lab actually still run with the ethos of helping, developing pipelines, methodologies, bringing technology, but sucking in other people's biological questions and working with those? Is that how you've developed the academic lab, or is it you have your own singular biological question and you're not really interested in that, but the collaborations?
No, that is again where it is actually hard to work like that. But I don't have my pet protein. I don't have my pet pathway. I'm a microscopist. I want to apply my microscopy skills to a wide variety of projects, and especially, of course, in the correlative microscopy field. So that is hard because there's not always funding for those kinds of positions. Yeah,
but you've got one.
I've got one, but again, I'm not part of the facility anymore. So when I got my promotion to professor, I was told I was too expensive to be heading a facility, and that still hurts me. But that's where nowadays you can have a pathway that is more or less technical, whereas that's now 7-8 years ago. That's where you had to go to, if you went that way, you had to go into an academic route. So that's where I was basically steered. I was always on a really technical path, and then I was diverted to a more academic role.
I know you said that still hurts, but it's from the outside, it looks like you kind of got the best of both worlds. So you don't have to worry about the instrumentation, and you can concentrate, as you just said, being in the lab, working on new scientific problems, not having your own pet protein or biological question to solve, but being an applied microscopist in an academic world. It kind of sounds pretty cool.
Yeah, in that sense, I'm probably I'm quite okay. I'm privileged.
I think that I bet there's a lot of core staff that are thinking, oh my God, that's actually what I'd like to do. Not to worry about the finances, the services, but you've got to worry about the grant funding.
Yeah.
I would argue, compared to many of your peers, because you are working with so many collaborators, your funding is coming in from all sorts of different avenues that if you were an individual, you're probably more resilient and more spread.
But you are also dependent on other people, which is not the fun part.
Yeah, okay.
That's where, and I have very good collaborators that they will always support that, but So making it clear also for new collaborations that I can't do all the experiments they want to do. I need people in my lab to do those experiments, so that if you apply for a grant, there needs to be stuff, personnel, but also consumables and things like that on that grant to support that.
Yeah, and yeah, as a collaborator, generally the academic, the lead PI is less likely to put. I think it's getting a lot better. You know, when you collaborate, collaborators are actually braver, more confident in putting on those extra costs for their collaborators to actually get the science done. And the grant panels are pretty wise that it does take that resource, and it is team science, isn't it?
Yes. No, that's what I thrive on, is working with other people, having those also especially interdisciplinary is quite cool. Working with physicists, with chemists, and things like that.
Thinking of your team,
yeah.
It's another picture for those who are listening. Who's the picture of, Paul?
So on the right, that is Lorna Hodgson, and in the middle is Judith. Judith has been with me since I moved to Bristol, and Lorna has been a postdoc in the group for at least 7 years. She's now moved into the facility. So that's where, again, as with grant funding, you can't guarantee that she'll be funded for the next couple of years. So that's where she took the decision, I will go into the facility, which is very useful for the facility. And this was to celebrate one of the successes we had as a group, where we worked with a guy called Jeremy Carlton, Jazz. And by chance, he was giving another seminar here today. And this was where this is one of the nicest correlative microscopy projects we've done, was published in Nature. So it really had an impact. And that was such a cool collaboration. Yeah, I'm still really, really proud of that one.
I just like the picture. It showed the group and the team efforts that behind. And as you said, you know, we as a team achieved a lot of those successes together, along with the collaborators as well.
Yeah.
What's been the hardest time in your career to date? Not saying you're going to get harder problems going forward. I don't wish that on you.
Well,
what's the most difficult time you've had to deal with?
Well, that was that time when I got professorship, which in itself was great. But that's where basically I was told, you're not going to lead the EM facility anymore. And that really hurt me, because we had just had a review of the facilities, and the last sentence in that report was actually, we should continue with having 2 facility heads, one for LM and one for EM. And then the next time I had a discussion with the dean, and he told me, we think it's good if you would step down as facility head. And that really hurt me, and basically where my baby was taken away from me. Because I together with Mark Jepson, we set up the Wolfson Bioimaging Facility, probably one of the very first really integrated light and electron microscopy facilities that was there, which I developed over the year. That's why at that point the facility was well known for as well, is the correlative microscopy. And that's where they asked me to step down, or told me to step down.
Yeah, so from the outside world, Paul, I would say it was never seen that it was taken off you, just that you had matured into a new position and could come in to look after your baby and let you carry on flourishing on the academic side of it. So from the outside, it was seen as a very positive and successful step. Yeah,
no, exactly. But it's also I'm used to the British system now, keeping up appearances.
Do you see it any differently now? Do you see it that actually?
No. That's where it takes time to realize that actually, a lot of the worrying things like recharging and the constant pressure from faculty management to make the facility break even and things like that. I don't have to worry about that anymore, and that's that's great.
So would you say it's been a good move for you now? Did they help? Did they do a good thing for you?
Come on. Yeah, probably. It's still hard. It's where there are times where I think, no, I want to be involved in that.
Yeah, I can see the influence. And do you know what?
Yeah.
Actually, it must be really hard for the person who stepped in, so Mark, in this case, it stepped in, to have you in the wings still. That must be, actually, I'd love to know what Mark's take is on this, because it's like, oh my goodness, but everything I do, I'm going to be judged by Paul. And if I do something that Paul doesn't like, it's going to look really bad. And because he inherited some of your team, I presume, as well.
Yep.
No, gosh, actually, I'd say he's probably got the worst.
Oh, yeah, no, I do realize that. And I don't envy him, because since that time, the financial pressure has only grown. So that's I don't envy him at all.
So that was a difficult time in your career.
Yeah.
What about on the flip side? In all your career, when was the best years? What was the best time?
I think the moves I've made. So after I did my PhD, I went to EMBL in Heidelberg. I was postdoc there for 5 years. And then at some time, my supervisor, Kai Simmons, he asked me, I'm going to set up a new Max Planck Institute. Would you like to help set up the new EM facility? And I'm like, yeah, that's a unique opportunity to really establish an EM facility completely from the start. And that's when also just designing labs, having the ideas of this is how I think it should look like. Those are really exciting times. And yeah.
So that was one baby.
Yeah.
And you left that baby to do the baby. But so what's your next baby going to be?
Yeah, that's what ideally, we would like to, because the technology is becoming more and more complex, and you really have to use the right microscope for certain biological questions. But you don't always have that microscope. So that's where I think we should go to a system where there is kind of a national facility for correlative multimodal imaging. That is what would be what we should be working towards, and what that's going to look like. There's been discussions going on. That's all, ongoing.
I would counter that. I would not look to set up such a singular resource. I think that sort of problem exists across the board in many other technology platforms, and to actually have a complete facility that is actually it's almost like a sabbatical facility where people just come, because that's where all the high-end new equipment, novel equipment is. And some of the more turnkey stuff, but we support access, because not every university. But your microscopes are £1 million plus. Your multi-beam SEM is, I've chopped onions and not wept as much as the price of some of these microscopes can be.
Yep.
And I actually think, but the same for mass spectrometry. Genomics.
No, exactly. So
evolving, and to have all these put in with the experts and scientists coming, meeting each other, and then going back. For me, so it's even bigger vision. So
it is bigger. That's why I didn't call it a correlative light electron microscopy national facility. It's a correlative multimodal. It is bringing more technologies together, where ideally you would want to go to a system where you have a molecule and you can place it in a cell, in a tissue, in an organism.
I love the idea. It's an idea that I've talked to multiple people about in the past. Yeah. It's a difficult world in any country because the way that the academic world works to make a neutral site.
Yeah.
That everyone and wanting and not to be hierarchical or territorial about it. But we'll talk after because I have ideas on that as well. So we'll talk afterwards a bit about it. I did wonder, besides that, whether your baby would be actually not the microscopes anymore, but the community, and how you're putting the communities together.
That's always been one of my big drivers, I guess. It's where you can do things on your own, but if you discuss it and bring people together, you can get much more done. And we don't need to reinvent the wheel.
No,
so that's why I've been very interested and very active in a lot of the community efforts, like Bioimaging UK, but together with Pippa Hawes, we set up EM UK, really, which was really designed for people doing electron microscopy, but interested in microscopy, but also the industrial people, bring those together and not have them separated.
Yeah.
And so that's where we're now. We've been very lucky. So there was this Chan Zuckerberg initiative for community building. And actually for the two communities I'm involved with, correlative microscopy, volume electron microscopy, both of those initiatives were funded. So we've just recruited a community officer for the volume electron microscopy community that is really now helping us, because so far that was all done on a voluntary basis, as you know. That is, and that's where I'd like to spend time on that, but I don't always have time to spend on it.
And you sent me this picture.
Oh, yeah.
Kind of.
Yeah.
I go. So go on, you go. Who's through this picture? Starting on
the left. Left is Lucy Collinson. In the middle is Kedar Narajan. And then we have Yannick Schwab. And at the end is me, as you can see. So one of the things that was always, I don't know, somehow on my bucket list is I want to organize a Gordon Research Conference. That's one of the best conferences I'd always been to, and that's something I would like to do. So with the four of us, we applied to organize a Gordon conference on volume electron microscopy. And with Lucy, we've been doing a lot of stuff already on volume electron microscopy in the UK. So that's where we made the decision, okay, Lucy and I are going to organize that first Gordon conference, which happened in July this year in Ventura in the US. And that was such an amazing meeting where you have all the experts there. It's in a somewhat secluded setting, so you can't escape each other as such. And then Yannick and Kedar were already nominated to be the next captains, in this case, of the next.
They've got captains' caps on in the picture.
Yes, that's what we got them at the end. So they will take over.
It's cool because I think actually everyone's been on a different series of the Microscopists, different seasons. So for every season, I think I've got one of you.
Yeah, no, these are my heroes as well. So it's I was very happy to do that.
Thinking of your heroes, have there been any standout inspirational people in your career?
Yes, that was my professor when I did my PhD. He's Ari Verkade, where at that time, I don't know if you had the same. When I was doing my PhD, I took electron microscopy pictures. I never was really completely happy with them. And then he really convinced me that this is world-class work. This is really very, very good. And he
he really motivated me, and he's been the biggest inspiration, I think, for during my career, where I've had a lot of supervisors, and you always learn from people. You learn the good things, but also the bad things. This is not how I want to be. This is what I do like about certain styles. Yeah.
And how familiar is he with your career now?
He's unfortunately passed away already 10 years ago from cancer. And that's happened to quite a few of my supervisors, unfortunately.
Yeah. Don't work with electron microscopy reagents. There's a lesson here.
Yeah. Maybe I should check myself. It's where, no, some of them were not electron microscopists as well. Yeah. They were just scientists.
Yeah, and age catches up with everyone at some point. Yeah. But he must have been aware of your career.
Yeah, no, definitely. And but also so I had two what they call promoters. So that's two professors that supervised me during my project. The other one is still very much alive, and he really follows what I've been doing. So he organized a gathering of his former PhD students when he had about 100 PhD students. That's why he said, okay, I need to get a party together. And he really knew what I was doing at that time. So he had followed up. Yeah. I was really happy about that.
So to move away from a slightly sad note, some quick fire questions. Yes, change our brain sets through it. Are you an early bird or a night owl?
Can be both. I used to be a night owl. Age is catching up. And so that's where it's also annoying, because in the weekend, I can't lie in anymore until 10, 11. I wake up at 8, 9 and just have to get out.
Why would you want to be in bed at 10 or 11?
From my memory, it was lovely to lie in bed until... That's why I'm very envious of my boys.
Yeah, except for when being a teenager, I think after the early 20s, that no, there's too much you can do in a day. Yes,
there is, yes.
A work day and a weekend as well. So, are you a PC or Mac person? PC or Mac?
Mac.
Mac? McDonald's or Burger King?
BK.
Okay. What's your go-to at Burger King?
I love the chili bites, chili cheese bites. Okay. So not a specific burger, but I always have the chili cheese bites.
Yeah,
if we go there. Yeah.
Germany or UK?
Difficult one. They both have their ups and downs. At the moment, I would say UK, because every time we go back to either Germany or the Netherlands, they're much harsher. That in that sense, UK really is a friendly country.
In what way?
If you bump into someone, and it's my fault, that other person will say, "I'm sorry" as well.
Yeah.
It's those little things where if you go into a shop, they nicely ask you, "Can I help you?" And if you say, "I'm just browsing," they will leave you alone. Whereas in those countries, that's very generalization, of course, but it is different.
So I was also going to ask UK or Netherlands, but I think you probably just...
Oh yeah, no, the Netherlands is probably even worse. I've always felt I wanted to go back, but every time we are then visiting family, we're shocked.
I've got friends in the Netherlands, so I visit the Netherlands. Not infrequently, and yes, the language is very direct.
Yes,
very direct. There's no wraparound. There's no P's and Q's. But I've always found it really friendly.
Yes, And I don't know if that is the case, but of course I speak Dutch.
I'm about to say, I'm not listening to the language.
Yeah.
I'm reading body language. I'm not hearing what they're saying, am I? You're right. Besides being able to count and say a few words, my Dutch is non-existent.
Yeah.
Very good point. Tea or coffee?
Coffee. Yeah.
Short or long?
Large. And I've only recently switched to oat milk rather than normal milk.
Okay. Wine or beer?
I'm not a real beer person. Can I choose cider?
Oh, you can choose cider. Actually, we're
Bristol.
I missed a meeting, so this is going to be very this moment in time for anyone listening to the podcast. But I missed a meeting last week, so I was over in Portugal talking, and we're on a multi-bid collaboration at the moment,
Yes.
And that has just been named cider. Was that your name?
Yes, it was.
I had a meeting this week with my internal collaborator, Joanna. I said, oh, it's cider, because one of the committee thought they really like cider. I thought, I bet I know who that is. That's got to come from Bristol. And so now you've just confirmed that. I love it. So, okay, sorry.
Yeah, no, exactly. But that's where you have to come up with these acronyms, especially for EU projects. That the acronyms are sometimes even more important than the content.
Wait, but it does sell a vision, and it does sell not the word, but when you know the acronym, it does sell the vision quite nicely. Chocolate or cheese?
Chocolate.
Milk or dark?
Milk.
Cadbury's or other?
Sorry?
Cadbury's or other?
No other. Cadbury's is way too sweet.
Okay. So what's your go-to milk chocolate?
You can basically the standard little one, but of course the Tony Chocolonely is very popular at the moment.
Yeah.
What you probably don't know, and I don't have it here, Verkade's chocolate. That's the best one.
Okay. So, well, we will be seeing each other at some point soon. You bring that, and I'll bring. Well, I can't bring Cadbury's.
No.
If that'd be wrong. I'll find something.
Yeah. No, that's where Verkade is actually a very famous chocolate and cookie brand in the Netherlands. So that's I like that.
You obviously have a slight bias towards it as well, of course.
Yeah, probably.
So what's your favourite food? If you were to be taken out somewhere, wine and dined, what would be the favourite meal on a menu that you could pick from?
A favourite dish would be scallops.
Okay.
I do like fish, but I do like steak as well. I'm not particular. Yeah, fish, steak.
Okay.
Those kind of.
Anything you don't like? What would be your worst dish? Your food nightmare? Cadbury's chocolate besides.
Yeah, it's something that's very bitter. I don't like bitter that much.
Okay. Yeah, I would go with that. That's quite a good answer, actually. Yes.
Yeah, chicory, for instance, is not my particular favorite.
So, if you're at home, do you cook?
Yeah.
You enjoy cooking?
It depends what time I get home, but definitely in the weekend, I try to cook. Yeah, and especially also since lockdown, I've started cooking much more.
I like the way you say you try to cook. Does that mean you were dreadful, or you just like to cook?
No. I've improved substantially, and I think that I get compliments, so it must be okay.
And I was going to say, so you do cook occasionally. What's your favorite film?
I do like the Star Wars, those kinds of Lord of the Rings.
So there is me going to ask next, Star Wars or Star Trek, and you've already answered it by saying Star Wars.
Yeah.
Okay. And what about your favorite Christmas film?
Oh, what's his name? Hugh Grant and
Love Actually.
Love Actually. I still cry with that.
Don't think I still cry with it, but I still watch it every year.
Yep.
Sure. And I've got to get out of the way of this picture. This is your family, I presume.
Yep. That is on the left is Guus. So he's studying mechanical engineering in Manchester. Next to him is Wouter. He is still doing his A-levels. And on the right is Joka. That's my wife.
And how old are they? Sorry? How old are your two boys?
Guus is 21 and Wouter is turning 18 in 3 weeks' time. It's really strange, but it's so this was at a Muse concert earlier this year, and that's why I used to go to concerts a lot because I love music, and over the years that I started doing that less and less, and then at some point I mentioned that I would love to see Muse. I haven't seen Muse yet, and I completely forgot about that. And then Wouter had remembered, and he told Joka, we need to get tickets for Muse and go and see them. So I really love that he remembered that is the thing I wanted to do. Yeah.
That's super cool. I've got to be careful. If I put my head here, it looks like I've got a new family.
Yeah, exactly. Yeah.
And final quickfire question. Actually, what music genre are you into?
Rock, progressive rock. So Muse, Rush used to be my favorite band. That kind of, I've seen Iron Maiden, Metallica, and things like that as well. But I've also seen Faithless, which I really enjoy. So I have a broad range of music. It depends on the mood as well.
I like it as you go to Iron Maiden, you get progressively heavier and heavier. So you're into your heavy metal with your music as well as in your staining of your samples.
Yeah, no, exactly. Heavy metal used to be really the thing I was completely into. Yeah.
And what's your favorite color?
Green.
Even though everything you look at is black and white, you like green.
Yeah, let's go for GFP. No, not that bright green, but.
So we've talked about some of the favorite times. We've talked about some of the difficult times as well, and the challenges. What do you do outside of work to chill out and relax? Have you got any hobby? What are your hobbies?
I don't have any particular hobbies.
You're worried about that when you retire.
Yeah, listening to music is something I still enjoy. And then what we do a lot in the weekend is we go to our allotment. So I used to hate the allotment. I never liked it. But then again, during lockdown, that's where we changed our plot as well. And that's where I started to appreciate. And so a lot of the food we eat is now our own homegrown food.
My aunt used to have an allotment. I used to do bits on it, and it was fun. But no, I can't commit time to that. I'd struggle. How often do you go to your allotment?
Well, during the summer, we used to go every weekend, or maybe both Saturday and Sunday.
Wow!
Because there is usually a lot to do. Lots of weeding as well, which is the annoying part. But yeah.
Well, it sounds excellent. I wonder how many others have got allotments. It's a question I've never asked. I'm going to have to work on that.
That's we had EMUK earlier this year, and all of a sudden we were talking about bushes that were growing alongside allotments. It had nothing to do with EM, but the discussion was diverted from that because apparently a lot of people had allotments and they all had their story to share.
EM UK, you need that as maybe one of the survey questions. Do you have an allotment or not?
Yes, exactly.
The proportion that's there. Because it's not just relaxing out of work, you can relax at work to a degree as well. And you sent this wonderful picture, which where was this? This is a lovely sunset.
This is a sunset. This is in Perth in Australia. So I think that's where we as scientists are quite lucky as well, that we get to travel and to see quite a lot of places. So this is in Australia earlier this year, where you get invited to certain meetings. This was the Australian Microscopy meeting. I did a review for Microscopy Australia as well, and it's just wonderful. But I usually spend too little time sort of outside of what we're supposed to do there. But this was one of the nice occasions. Yeah.
And so the other side, the bit we haven't really talked about, we've talked about communities, we talked about your science, the facilities, going to the academic career. But you started very early on talking about how you want to be a teacher, and you are still arguably a teacher, and you do a lot of courses.
Yes.
So obviously based around your speciality, so CLEM courses such as this. So how many? I bet you'll never be able to give an answer. How many courses do you think you've taught on?
Must be 40. I don't know. No.
Yeah, a lot though.
A lot. Yes. Yeah.
And if you look at this classroom size, you sent a picture, and there must be probably 40, probably in the classroom as well.
Yeah. So this was just before lockdown. This is the very last thing I did before lockdown. This is in Portugal with Erin Trentfield, and this was again, it's a CLEM school, and really, Erin uses this term MacGyver. So not everybody has all the sophisticated tools you can do correlative microscopy with, but basically what we taught in that course is even with the basic tools, you can do very nice and very good correlative microscopy. I mentioned that collaboration with Jez Carlton, which got a Nature paper where we celebrated. That was done with these special imaging dishes, just a confocal imaging dish, just an electron microscope. That was the whole idea of getting that message across, that you don't need to do it always that hard and with all the biggest tools you have.
Yeah, I think so. So Erin uses MacGyver. I think I'd go Heath Robinson as another example of. We've used sticky tape, glue tape,
yeah.
All sorts of improvisations to get things to work.
And that's where these in-person courses are so valuable, because that's where you can show all those minute details, all the little tips and tricks, which are much harder to show when you're doing that, let's say via Zoom or things like that.
And actually, I'd say arguably you're helping to encourage people to think about tips and tricks, because when I guess we're fairly similar age-wise, I'm guessing.
Yep.
And when
I know how old you are.
I'm 50, Paul.
Okay, then you can add 9 to mine.
You're 59.
I am 59.
This is a complete digression to where I was going. Oh my God, you're young for 59.
Thank you.
And that's not just your camera, because I know you in person. Oh my God. Yeah. Well, see, you can't leave me speechless.
No.
You're the first guest to leave me speechless. You rotter.
Yeah.
Damn it. So you will remember the days. Back on track. I can't believe that. back on track with this. I think we were at a generation of going through, and I think all the generations before us, where things didn't necessarily, let's take this off, necessarily work perfectly. And we were used to technology being difficult to operate, computers crashing regularly when you're taking your images, crashing, your time lapse crashing, having to improvise around it. But credit to the manufacturers and all the companies out there. They've made things a lot more turnkey so the biologist can get on with the biology.
Yeah.
But now we're noticing when something goes wrong, there's not as much determination to solve the problem, but just find another solution or another question that they can answer easily, rather than to persevere, troubleshoot, and get there in a meaningful way. That there seems to be a trend at the moment towards ease of use, rather than actually, I'm not going to get to answer my question unless I, like you did with your impact, creating something to solve the problem. I think there's fewer of that nature. It's not, again, stereotyping to a degree. So I think your tips and tricks encourages that mentality to solve the problem, create a pragmatic solution.
Yeah, no, exactly. And that's where, it's always the biological question that should drive the technology, so that you need to use the right technology to answer a question. So don't use, I don't know, let's say cryo-EM because you need to count the number of mitochondria in a cell. That those two don't match up. But it's sometimes where there are these buzz words, technologies that people have heard about, seen in a conference, and they have to use it.
So I think that's good advice. And actually, of course, you did send me one other picture. And
yes.
When I looked at the post, I realised that was me at the front with you on a course. Was this in Bristol?
This was in Bristol. So that's on one of the EMBO courses on correlative microscopy. I don't know the exact date, but we were both a bit younger there as well.
Yeah, most definitely. Proportionally, I was a lot younger though. I've just found out.
Yeah.
I have one other. Actually, I have two other questions, so we've got to be careful because it's really close to time. We've talked about all the developments, and one question I haven't asked. You developed the CLEM pipeline. How difficult was it? How difficult is it still getting some of your peers around you, researchers with biological questions, to embrace that technology and realize it can help them solve a question?
Yeah, it is still an issue that they will. You first need to show a very good example, a proof of concept study that really has made that impact. So that's where, for instance, again, that study with Jess Carlton, that really changed a lot about how people perceive that CLEM workflow. That is, I think that's still hard sometimes.
Yeah, and I guess as a technologist, and I'll come on calling you a technologist now, because I think you are a technologist. How difficult, you must have come to terms with it now and understand it, but how difficult did you find it coming to terms that people weren't wanting to jump on this and try it because, oh my goodness, it's going to let them do stuff they couldn't do before, but they were just like, no, not interested.
Yeah. Oh, there are enough other people that are interested and that do, I don't know, see the advantage of using certain technologies. That I'm not too bothered about. So if someone thinks, oh no, that's nothing for me, or that's not doing it for me.
Is that a matured thought? Was it like that 15, 20 years ago?
Oh, probably I was a bit more angry young man. It's
well, no, not that young then.
I was probably
not trying to, I don't know, sell the technology as much as I'm doing now. And now I'm much more an advocate of the technologies. I was probably more focusing on my own, so to say, technology development. And that's probably, one of the things you do have to think about. Okay, it's nice to do all the technology development, but what are you going to do with it? You do need to have that biological question in the background.
The final two really quick questions, because we are on the hour. Do you have any regrets scientifically?
I could have taken other career options, but do I regret that? No.
Which is cool, isn't it?
Yeah. No, and I'm happy with the way things went. Yeah.
And just look at your friends, work friends. How supportive they are. That picture that you sent, the four of you. They're not just work colleagues.
No, they're my friends. That's yeah.
Yeah, I think that's, again, science is a very lucky community in that respect.
Yeah.
That you make friends even outside your own workplace. In fact, sometimes closer friends outside your workplace than you do in the workplace.
Yeah.
Which is probably a good thing, because you're living together in work and not outside of work to the same degree. Yeah. And if you could, so I didn't ask you this question earlier, I'd like to ask it. If you could do any job for a day or a week, just to sample what it'd be like to have that type of career or in that type of environment, what would it be?
Dive instructor.
A
dive instructor.
A dive instructor. That's I think we might have had ships, we've had astronauts, but a diving instructor, that's pretty cool.
Because I used to, when we was at EML, we had a diving club and we went a lot of diving, and there was within the B sector, the British Sub-Aqua Club Association, there it was really easy to become an instructor. So I was a trainer. I've dove for many years, and I would love to go back and do that. But also, if you look at those dudes that are the dive instructors, they're the cool guys, which I never got to be.
And do you dive at all now?
No. I would love to do that again, but yeah. It's always like, what's holding you back? I don't know. There's always other things to do as well.
I will say, Paul's boys, you got a Muse concert. Now you know what to do for next Christmas.
I'll definitely show them this movie.
Oh my God, you're going to be 60. I just thought. You said you're going to be 59. You can have a diving party.
Yeah.
Birthday.
That's only next year.
That's fine. You've got time to arrange it.
Yep.
Paul, we are gone the hour. So everyone who's listened today, thank you for listening. Please don't forget to subscribe to the channels. And you saw pictures earlier of Yannick, Kedar, Lucy, all have been guests. Go back, listen to them. They all talk about Paul during their talks as well. And obviously the Volume EM community that was with Lucy and with Kirk. So again, another very useful one to listen to, and many others aside. But Paul, thank you so much for joining me today. You've been a blast. And I've learned that you're 59.
Yeah. Thank you so much for having me. It was great fun. I really was thinking, what are we going to talk about during this? And we've covered a lot. Yeah.
So, Paul, everyone who's listened, thank you very much.
Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.